Chapter 4, Specimen Collection and Transport. Contents in this chapter: Equipment, Gloves, Goggles, Antiseptics, Tourniquets, Collection Tubes, Syringes, Needles, Disposal Unit, Centrifuge, Labels, Barcodes, Identification Tags, Requisition Forms, Order of Draw, Uses of Collection Tubes, Specimen Transport, Procedural Complications, Allergic Reaction, Bleeding does not stop, Fainting, Hematoma, Hemolysis, Hemoconcentration, Indwelling limes or catheters, Infections, Prolonged tourniquet application, Equipment. The following equipment is commonly used in phlebotomy: gloves. Disposable gloves are used in phlebotomy to help prevent contamination. Latex, nitrile rubber, neoprene, or vinyl. Two types of gloves: exam gloves and surgical gloves. Surgical gloves are better sized and made to a higher standard. Goggles, although not common, but more frequently recommended for use. Protect eyes from exposure due to splashing of blood-borne pathogens. Antiseptics. Antimicrobial substances are used to clean puncture sites before the procedure in order to reduce possible infection. The most common antiseptic is alcohol, tourniquets. Compressing device that is used to control blood circulation. Usually rubber tube-like, that is wrapped around the arm during the procedure. More modern devices are available with contour cuffs attached to the wire. Collection tubes. Made of glass or plastic, plastic being preferred in use. Tubes are attached to a needle and a hub. Contain vacuum so that tubes fill automatically to a certain level. Some tubes contain anticoagulants, or additives. Remember, anticoagulants are substances that prevent blood clotting. Heparin is the preferred anticoagulant in clinical chemistry. Lithium heparin is the general anticoagulant to study glucose levels. Acid citrate dextrose is an anticoagulant used in blood donations. In tubes with anticoagulants, it is important to fill the tube with the correct amount of blood to provide an accurate blood additive ratio. Syringes. Medical syringes are usually fitted with needles. Consists of a plunger and a tube. Plunger fits exactly into the tube. The design makes it possible for fluid to be pulled in and pushed out the syringe. Needles. Needle sizes vary as do the gauge, opening, of needles. Larger gauge sizes refer to smaller openings, diameter, of the needle. The butterfly needle refers to a winged needle, infusion, set. Butterfly needles are used when conventional methods are too difficult. Needles have a color coding which refers to the gauge size. 19 gauge, white top. 20 gauge, yellow top. 21 gauge, green top. 21 gauge butterfly needle, green top, often used in pediatrics. 22 gauge, black top. 23 gauge, blue top. 23 gauge butterfly needle, blue top. 24 gauge, purple top. 25 gauge, orange top. 25 gauge butterfly needle, orange top. Disposal unit, also known as a sharps container. Medical needles and other sharp objects/instruments are disposed into it. There are two different types. Single-use container will be disposed of with contents inside. And reusable, automatically emptied and sterilized for safe reuse. Centrifuge. Device or machine with a fast rotating container that applies a radial force. Used to separate fluids of different densities, as in spinning blood. If blood has a separation device, it should be centrifuged once. Labels. Small pieces of paper or plastic attached to the collection tubes or containers. Labeling collected specimen or tubes must be accurate. Elements of labels are patient's first, middle, and surname. This must match requisition form. Patient's ID number. This must match requisition form. Date and time of collection. Initials of the phlebotomist. Automated systems may have labels use barcodes. This is more reliable. Barcodes. Codes that are machine readable. Usually consists of numbers and patterns of lines printed on labels. May be used for patient names, ID numbers, and date of birth. Specimen accession numbers. Test codes. Product numbers. Expiration dates of inventory. Handwritten information can't be converted into barcodes. Identification tags. Small tag, usually worn on bracelets, with information on medical conditions. Tags allow for fast medical attention in emergencies. Newer forms of ID tags are USB alert tags, capable of holding more information. Other forms of ID tag are RFID tags. RFID tags use silicon chips and wireless receivers. Requisition forms. Forms to properly identify blood samples. Each specimen collected needs to be accompanied by a requisition form. Elements of the requisition form are patient's name, ID number, date of birth, and gender. Name of physician requesting procedure and tests requested. Source of the specimen, if analysis and reporting are site-specific. Date and time of collection and initials of the phlebotomist. Report forms, clinical chemistry, may also include reference ranges, order of draw. In order to avoid cross-contamination, blood collection tubes must be drawn in a specific order. Alkaline phosphate should never be collected in gray top tubes. Lead levels should be collected in tan top tubes. Copper should be collected in dark, royal, blue top tubes, no anticoagulants. Blood cell count in purple top tubes. Cytogenetic analysis in green top tubes, yellow top tube blood cultures, SPS sodium polyanethyl sulfonate, mix by inverting 8 to 10 times. Light blue top tube citrate tube mix by inverting 3-4 times. Gold top tube gel separator tube mix by inverting 5 times. Red top tube serum tube mix by inverting 5 times, plastic. Glass tubes require no mixing. Orange top tube rapid serum tube mix by inverting 5-6 times. Green top tube heparin tube mix by inverting 8-10 times. Purple top tube EDTA tube mix by inverting 8-10 times. White top tube separator tube with gel mix by inverting 8-10 times. Gray top tube fluoride, glucose, tube mix by inverting 8-10 times. Uses of collection tubes. Yellow, black top tubes. Additive, broth mixture. Mode of action, preserves viability of microorganisms. Uses, microbiology. Light blue top tubes. Additive, sodium citrate. Mode of action, forms calcium salts to remove calcium. Uses, coagulation tests, full drawer required. Red top tubes. Additive, none. Mode of action, blood clots. The serum is separated due to centrifugation. Gold top tubes. Additive, none. Mode of action, serum separator tube. Contains gel at the bottom to separate blood from serum due to centrifugation. Uses, immunology, serology, chemistries. Light green top tubes. Additive, plasma separating tube with lithium heparin. Mode of action, anticoagulants with lithium heparin plasma are separated. Uses, chemistries, dark green top tubes. Additive, sodium heparin or lithium heparin. Mode of action, inactivates thrombin and thromboplastin. Uses, for ammonia level use sodium or lithium heparin. For lithium levels use sodium heparin. Purple top tubes, lavender. Additive, etiliquid. Mode of action, forms calcium salts to remove calcium. Uses, blood bank cross-matching and hematology, CBC. Requires full draw. White top tubes. Additive, potassium EDTA. Mode of action, forms calcium salts. Uses, molecular, PCR and DNA testing. Pink top tubes. Additive, potassium EDTA. Mode of action, forms calcium salts. Uses, immunohematology, gray top tubes. Additive, potassium oxalate and sodium fluoride. Mode of action, antiglycolytic agent preserves glucose levels for up to 5 days. Uses, glucose testing requires full draw, short draw may lead to hemolysis. Light brown top tubes, tan tube. Additive, sodium heparin. Mode of action, inactivates thrombin and thromboplastin. Contains virtually no lead. Uses, serum lead determination. Black top tubes. Additive, buffered sodium citrate. Mode of action, forms calcium salts to remove calcium. Uses, sedimentation rate, requires a full draw. Yellow top tubes. Additive, ACD, acid citrate dextrose. Mode of action, complement inactivation. Uses, paternity testing, DNA studies, HLA tissue typing, specimen transport. Specimens that are collected need to be transported to laboratories for further testing. Certain regulations that apply. All specimens must be placed in leak-proof containers, tubes. Tubes must be bagged in special biohazard specimen bags. Requisition slip must be placed on the outside pocket of the bag. One of the safest and most efficient methods of transport is the pneumatic tube system. Transport for tube specimen. Tube specimens can be sent using the pneumatic tube system. Pneumatic tube system. Place tubes in a biohazard bag and seal. Attach requisition slip. Place biohazard bag in the pouch. Completely seal pouch. Load pouch into the pneumatic tube and send to the lab. Certain specimens need to be transported to the lab more quickly in order to get accurate testing results. Blood and urine samples must be transported quickly in order to increase the chance of pathogen detection. Serum specimens should be transported to the lab within 2 hours in order to avoid erroneous results. A delay or improper transport may cause hemolyzed specimens, which can compromise laboratory testing and/or assays. The specimen to be kept cold during transport is by using icy water. Blood gases. Gastrin. Ammonia. Lactic acid. Catecholamine. Renin and parathyroid hormone determinations. In the case of light-sensitive specimens, it is best to wrap the collection tube in aluminum foil to prevent exposure to light. Bilirubin. Beta-carotene and erythrocyte protoporphyrin, procedural complications. The following are potential complications a phlebotomist may encounter while performing routine procedures, as well as the proper preventative measures and responses to these situations. Allergic reaction. Some patients may be allergic to supplies used during puncture such as latex gloves. Patient records should always be checked for known allergies. If the patient has allergic reactions that were unknown, be prepared to provide or call for medical attention. Bleeding does not stop. In some cases, such as patients with bleeding disorders, that the bleeding does not stop. If this occurs, the patient should be instructed to lift the arm above the shoulder and apply pressure. If bleeding does not stop after 10 minutes, contact the supervisor. Patients, where bleeding does not stop, should not be left alone until the bleeding stopped. Fainting. If the patient is known to faint, the patient should be laid down for the procedure. If the patient faints, the procedure should be stopped immediately. Patients' heads should be lowered to their knees. Keep patient in sight for 15 minutes before releasing him. Contact supervisor or physician if the patient is unresponsive. Hematoma. occurs as the blood that pools in the tissue around veins is collected. It may result in bruising and may be painful. To prevent hematoma, surface veins should be chosen and puncture should be at the accurate angle. Hemolysis, a complication that occurs in the collected blood sample. If the sample is not collected or handled properly, blood cells may start to break apart. To prevent this, the phlebotomist should collect and handle samples with care. Tubes that contain additives should be inverted. Hemoconcentration, the increased amount of large molecules and formed elements. Causes may be prolonged tourniquet application or occluded veins, indwelling limes or catheters. This can be a source of potential errors. To prevent complications, samples should be discarded 3 times the amount of line before the specimen is collected for further testing or analysis. Infection. When performing phlebotomy, the skin barrier is broken, allowing germs or bacteria to enter the body. To prevent infection, skin must properly be cleaned/sterilized and gloves should always be used. Prolonged tourniquet application. Tourniquets should not remain on the patient for more than 1 to 2 minutes. If left on longer, the integrity of blood and health of the patient may be at risk. Prolonged tourniquet applications may lead to tissue or nerve damage. If no vein can be found, the phlebotomist should try an alternative site.